Hello and welcome to today's webinar, the ABCs of ADCs, ovarian cancer, and antibody-drug conjugates. I'm Maggie Nicholas Alexander, the Senior Director of Gynecologic Cancer, Patient Support, and Education at SHARE. Before the presentation begins, I'd like to tell you a little bit about SHARE. SHARE is a national nonprofit that supports, educates, and empowers anyone diagnosed with breast or gynecologic cancer and provides outreach to the general public about signs and symptoms, because no one should have to face breast, ovarian, uterine, cervical, or metastatic breast cancer alone. For more information about our upcoming webinars, support groups, and helplines, please visit our website at sharecancersupport.org. And now we're very excited to have Dr. Alldredge joining us today as our speaker for this webinar. So I'm going to hand it over to her now to introduce herself.
Hi, I'm Jill Alldredge. I'm a gynecologic oncologist at the University of Colorado, where I've been for 5 years since finishing training out in California. I provide the full scope of gynecologic oncology care through the university, so that means doing all surgeries needed for gynecologic cancers, as well as prescribing chemotherapy, maintenance therapies, surveillance care, genetic testing for those patients. So really trying to provide comprehensive care across the board for individual patients. And then I work to enroll patients in clinical trials, and I work through several national organizations on trying to move the needle ahead for gynecologic care nationwide through clinical trial work. So I am Maggie. I am gonna try to share my slides here. Here we go. Thanks for your patience, everybody. Here we are.
All right,
Oh,
looks good.
perfect. So today I'm going to talk to you a little bit about antibody-drug conjugates within ovarian cancer. And I think that what we'll start with is just going over what is an antibody-drug conjugate, or ADC. What are some of the unique side effects for these? And what are some of the potential ways that we can use ADCs within the field of ovarian cancer, including some current FDA drug approvals? And then I'll tell you a little bit about what may be coming down the pipeline in the future. So just to get started, an antibody-drug conjugate, I think of as a Trojan horse. So basically what we're doing is we're linking an antibody that binds to the surface of the cells. And cells have little markers all over their surface, and this antibody links up with one of those surface markers. And that antibody is linked together with a little chemotherapy. And the idea is actually this really clever mechanism where this antibody attaches to the surface proteins on a cancer cell, and then that cell eats it up. And I think this bottom picture here shows it best, that it basically starts to kind of go in and it swallows up those surface markers, including the antibody with the linked chemo. And then it creates this little bubble that gets delivered onto the inside of the cell, where that chemo can then go have its action. So that's a really clever mechanism to try to decrease the toxicities of our traditional chemotherapy, whose job is really to go out into the system and kill all quickly dividing cells that it comes into contact with. So that works to kill cancer cells because they are quickly dividing, but it can also have side effects to other nearby tissues who happen to be quickly dividing. Things like our gut or our bone marrow and our blood counts. So that's where we get a lot of the side effects from chemo, is because chemo is killing cells that we don't really want it to. So this mechanism, this Trojan horse mechanism, really allows for chemo to get delivered into the inside of hopefully just the cancer cells. So this is working by taking only the markers, those surface markers that are highly expressed on the surface of cancers or tumor cells, and are not expressed or very lowly expressed on normal tissues. And so we can't use this for all cancer types, and we can't, make all chemos deliverable this way yet. But we're starting to find that especially in ovarian cancer, there are a handful of surface markers on top of ovarian cancer cells that we can take advantage of. So some of those are things like the folate receptor alpha, mesothelin receptors, TROP2 receptors, tissue factor, and NaPi2b. And I know those are just a lot of letters and numbers, and that part's not terribly important. The idea is that we're finding more and more that there are unique markers on the surface of ovarian cancer cells that are not expressed on normal tissues, and we can take advantage of that. For the moment, the chemotherapies that we link up to this antibody are mostly like Taxol. And many women who have been through ovarian cancer treatment have seen Taxol as one of their early on chemotherapies. And this is a really similar mechanism to that. So these are little chemos, MMAE or DM4, that are linked up with that antibody. So this has been an area where we have done many trials over the last few years trying to figure out which antigen or surface marker on the ovarian cancer cell we might be able to take advantage of. So as you can see here, many of these are highly expressed on the surfaces of ovarian cancer cells, and then they have a chemo link payload to them. And some of these have been not as promising as we wanted them to be. Phase 1 clinical trials are really early trials where we're trying things out, trying to find the right dose, just get a basic sense of whether they're going to work or not. And you can see these are some low percentages here for some of these different agents. But for some, we've had some really promising initial results. And we're going to talk about these in a little bit more detail. But the promising initial results have then led to further clinical trial development. And I know that this can look like a lot of jargon, so I just want to point out that the abbreviations here, ORR, means overall response. So that means people who had a complete resolution of all of their disease and people who had a partial resolution of their disease, so some shrinkage. So when we're looking at these percentages, these mean these women had some sort of response to these agents. PFS, when we talk about progression-free survival, really means the time between recurrences. And our goal with ovarian cancer is to get as long as we possibly can in between recurrences, since oftentimes complete cure can be difficult in these recurrence settings, achievable, but we typically aim for the longest duration as we can between recurrences, or PFS. So these potential targets have led us down the road of 3 drugs that I'll tell you a little bit more about. The first one, its brand name is Elahere. We call it Mirvetuximab or Mirv. So you'll see us talk about each of those 3 names interchangeably. The antibody for Elahere is a folate receptor alpha antibody. And you can see here in this picture, this is a highly expressed folate receptor alpha. So all this brown is folate receptors on the surface of these tumor cells. And that's linked to a Taxol-like chemotherapy. In the clinical trials that we've done in this space with mirvetuximab, those are not restricted to a certain subtype or histology of ovarian cancer, though they are for epithelial ovarian cancers. So not for sex cord stromal tumors, not for germ cell tumors. These are epithelial cancers. So things like high-grade serous, endometrioid, clear cell, carcinosarcoma, those are the types included in the trials. And that in the original study that we did in this space, I'll show you in just a moment, we really found that this isn't helpful to give to all women with recurrent ovarian cancer. It's really helpful and limited in its benefit to those who have more than 75% of their cells. So if we took a slide like this, more than 75% of those cells or expressing that folate receptor 1. So not for all recurrences. As of now, we don't have any data on giving these targeted folate receptor drugs in upfront ovarian cancer. So the standard of care remains a combination of surgery and carboplatin and Taxol or cytotoxic chemotherapy. And Elahere is not part of the upfront management yet. And I will say that perhaps that changes in the future, but we're not there yet. So the places we'll talk about today are in platinum-sensitive ovarian cancer and then an FDA approval in platinum-resistant ovarian cancer. So just to touch base on what those terms mean, so using Mirvetuximab in platinum-sensitive recurrent disease means that from the time someone saw their last platinum drug, usually carboplatin, it's been more than 6 months. That means that we have a platinum-sensitive type of tumor. And we did have a phase 2 trial done in just women with more than 75% folate receptor expression. This was called the Piccolo trial. And hot off the presses in June of this year was some really promising preliminary results out of that trial, saying more than 50% of patients had some sort of response, which when we compare that to just traditional chemotherapy in this space is better. So really exciting result hot off the presses here. And that the average time people stay in remission with that is about 8 months, which again is about the same as what we get out of traditional chemo in this space. So I do think that this will, with a little bit further data maturation, will end up being likely our second or third line choice in this setting. There is also some data ongoing about adding the drug Avastin to Elahere, and those results are also really promising and exciting that maybe the combination of those two drugs could be better than Elahere all alone. And so that's underway as we speak. That's the Gloriosa trial that's enrolling across the US and maybe a really wonderful trial to look into if it's open at an institution near you as we continue to develop this. We've also studied Mirvetuximab or Elahere in the space of platinum-resistant or refractory disease. And that's when someone has a recurrence less than 6 months from their last carboplatin drug, or that they've had progression of their cancer while on a carboplatin drug. And so this was where Mirvetuximab first came online. We did a trial called the Forward trial, where we found that really didn't seem any better than chemo. But when we looked under the hood and we looked a little bit closer, we found that we were looking at everyone, all amounts of folate receptor expression. And if we took all comers, we don't really have much benefit. But if we really look at those patients who have high expression, so more than 75% expression of folate, those women have a great response to this drug. So that led to another trial called Soraya, looking at just that group, showing good promise. And then a phase 3 confirmatory trial. That means it compared Elahere against the current standard of care, chemotherapies, and it showed that it was better than the current chemotherapies that we use in this space, and tended to have less side effects than traditional chemo in this space. So this really exciting data out of the Mirasol trial led to an FDA approval. So for right now, this is the only FDA approval of an antibody-drug conjugate in ovarian cancer, but I do think that more are likely to come, given all of this exciting information. Mirvetuximab is a unique drug in its side effects. So the linker that holds together the linker that binds up the immunoglobulin and the chemo has its own unique side effects. So that linker can cause blurry vision or dry eyes in about 60% of patients. Usually takes about a month for that to take effect. And for the vast majority of patients, that will resolve either completely or almost completely, but that takes some special care. We have to give special eye drops to try to prevent eye toxicities, and we have to have folks follow with an ophthalmologist throughout. In addition to that really unique side effect that comes from the linker, we can also just see some traditional chemo-like side effects. So some things just like Taxol, neuropathy, so numbness or tingling in the fingers or toes, maybe some hair thinning, some low blood counts, and then rarely some lung inflammation. So some unique things that come with this Elahere drug. Overall, though, tends to be easier to tolerate than chemotherapy is. I do want to briefly touch on a few other ADCs coming up in the ovarian cancer space. We have DXD is what we're calling it, raludotatug. It's a mouthful, but this is directed against CDH6, and it's linked up with a topoisomerase chemotherapy. And then we have several trials coming down the road in platinum-resistant ovarian cancer with some promising early results. Just to give reference, a response rate of almost 50% in platinum-resistant cancers, a really good control rate. So really promising. We're looking forward to more work in this. The other, we call it SAUCY, but sacituzumab is the formal name of this. And this has been so promising in early trials that it's got a fast track designation. So the FDA really wants this to be studied as quickly as we can. And this also really promising results. The linker in this one maybe causes some mouth irritation or mouth sores, more so than eye side effects. So we have several really exciting antibody drug conjugates. So a chemo linked up with a Trojan horse mechanism that are in development in clinical trials across the country. And if you go to clinicaltrials.gov, you can search for clinical trials available in your area, and many will include antibody drug conjugates, or talk with your doctor about whether this is something that makes sense for your cancer, or if there's a trial available for you. Antibody drug conjugates are really exciting and are a developing area that I think of as personalized medicine, right? We're asking what your tumor expresses on its surface and then giving you Trojan horse chemo to link up with what your cancer cell expresses, which I think is as personalized as we can get. That being said, it does have some unique side effects, and we have to be really cautious in people's care to make sure they're seeing eye specialists or taking special preventative measures to keep safe from those side effects. So I will turn it over to you all to share questions with me.
Great. Thank you so much for that presentation. That was a really great introduction to ADCs. So now let's start the Q&A. There were a lot of pre-submitted questions, and you can still submit questions in the Q&A section at the bottom of your screen. We will try to get through as many questions as we can in the remaining time. So side effects came up a lot in your presentation. And so one question is whether you think that the future ADCs are going to have a reduced side effect profile compared to Mirv.
When do I think?
What do you think?
Oh, do I think? I do think that antibody drug conjugates are relatively new in the world of oncology. They've really been up and coming in the past, I would say, 5 years or so. And so certainly we have seen in lots of drugs that given time, we can come up with small revisions, small changes to the structure, maybe of these linkers that are causing the trouble. And perhaps we could decrease that side effect risk in the future. I do think that just like with chemo, we have side effects on all quickly dividing tissue. When we're having side effects from the linker, it's usually that we have some expression of those things, like tissue factor can be in the eyes. And so that's where we can see some of the side effects of tissue factor ADCs. And so there's going to be some expression of these different surface markers and other tissues that I'm not sure we're going to be able to get around, as things develop. But certainly as these become more and more mainstream, I'm hopeful that the side effects can decrease.
Great, thank you. And I obviously Mirvetuximab has the full is for those who have a folate receptor alpha expression, but I think there's some confusion from our audience about some of the other targets. So could you maybe reiterate the ones that are not looking at that folate receptor alpha expression?
Mm-hmm. So a lot of those in the early trials are aiming, because a lot of those are phase 1 trial spaces still. We're still aiming for tumors that seem to highly express those markers. But for right now, in early phase trials, we'll often try them in all comers, just like we did with Elahere early on. In Elahere, we looked at everybody and saw no good responses. And then when we narrowed the field to just people with a high expression, that's where the benefit was. So we're going through similar paradigms in some of these other antibody drug conjugates, is looking at all comers, but also just having planned groups where we have high expression to make sure that we're not, I think for lack of a better phrase, we're not throwing the baby out with the bathwater, right? We're not saying, oh, well, this doesn't work for all comers, so it's garbage. Because if we had done that, we never would have had Elahere. So I think we have to plan to have these high expression groups because that's where we expect to have the greatest benefit.
Great, thank you. And then someone's asking about, they just started Mirv and, one of the side effects is neuropathy, and what's a good way to prevent that?
Yeah, so that neuropathy is coming from that chemo linked up with the antibody drug conjugate, that Taxol chemo. In general, for both Elahere with that Taxol and just for Taxol in general, there are lots of layers of options. So there can be things like vitamin B6 and B12 can sometimes be helpful. Using things like capsaicin creams, acupuncture, CBD oils or creams, depending on where in the country you live and what's available. And then there are medication managements out there as well. So I think there are lots of layers of things to manage neuropathy, and I would encourage you to talk with your doctor about what they can offer, or what a physical medicine and rehab specialist in chemoneuropathy might be able to offer, because I think there are lots of choices to help with that side effect.
Great. And then I know you mentioned that for Mirv, that it's epithelial ovarian cancer, so could this be appropriate for someone with low-grade serous who had that expression for
Tricky.
folate receptor alpha?
Yeah, I do think that low-grade serous is a unique subtype of cancer, but I would say in those that have high folate receptor alpha expression, yes, I would still expect this to be an option. I think that circumstance where a low-grade serous is highly expressing is a relatively rare beast. But I do think that I would think of this independent from the epithelial histology, and think of it as a treatment for folate receptor high tumors.
And then we're getting a question about how is this different from PARP inhibitors, essentially?
Oof. Yeah, so PARP inhibitors, that would be a whole lecture for you. But in a nutshell, PARP inhibitors are trying to take advantage of some of the broken DNA communication and repair mechanisms happening. So a faulty game of telephone tag that we're shutting down to kill off cells that are not communicating well and aren't listening to the normal checks and balances. So kind of a different mechanism than this one, which is, here's the cancer cell with a bunch of markers on it, and let's deliver chemo only into those cells, just to try to focus where chemo is being delivered. So mechanistically, kind of different beasts and should not be exclusionary to each other, right? So if we've had a PARP inhibitor, it doesn't mean we can't have MIRV. If we've had MIRV, doesn't mean we can't have a PARP inhibitor, because they are separate.
And then we have a question about, how does someone know if they have that folate receptor alpha expression?
Thank you so much for asking. I should have mentioned that. So more and more institutions, when you have your initial surgery, are actually adding this on to the pathology that comes out of that initial surgery. So when you have your initial surgery, you'll get a pathology report that says, it's this stage and it's in these places and it expresses estrogen or whatever else, and it expresses or does not express folate receptor and at what percentage. So many institutions have that built into their normal pathology infrastructure now, or your physician can ask for banked tissue, sort of tissue that your institution's held on from a prior biopsy or surgery, and they can ask for that to be added on to existing tissue. The other option is through molecular testing groups, things, I won't list all the different brand names, but there are lots of molecular tumor profiling services that we use, and this is one of the markers that those will report back on.
Great, thank you for that. And then, how long can someone be on MIRV? Is
So
there a time limit?
Yeah, great question. So both in the platinum sensitive and resistant spaces where mirvetuximab is currently has the most momentum, those are to be continued until progression or toxicity. So either until it stops working or it's too toxic to tolerate. So that can really go on for months, years, however long it's doing its job. In patients who have a complete response on Elahere, we'll often have a conversation about when makes sense for them to stop. But all of the clinical trials are built as progression or toxicity, not a set number of cycles.
Mm-hmm. And at what point should someone consider maybe going off of it, like in terms of the side effect, like how bad do the side effects essentially have to be that you think it's warranted to consider going off of it?
Yeah, so I do think that physicians prescribing this medication are keeping eyes out for things like bone marrow blood counts. They should be asking about neuropathy every visit. They're keeping an eye out for things like lung toxicities. And so they're watching for those things to help guide when we should stop. From an eye side effect standpoint, the ocular toxicities, we rely on our ophthalmologists to really help guide that. They're doing special eye exams every time you go to see if the external surface of your eye is really irritated or not. If you're having really bad dryness or eye pain, those are reasons to stop. So if side effects are too much or if it's not working, we should stop using it.
Mm-hmm. And what percentage of patients have a complete response with MIRV?
So I would have to say it depends a little bit on whether we're platinum sensitive or platinum resistant and how many other drugs we've had. But just to show you kind of generally, in platinum resistant patients, about 40% of patients have a response. That includes a partial and a complete response. And in platinum sensitive, about 50% have a response. That includes partial and complete responses. So higher than traditional chemo in both of these circumstances, which are typically somewhere in the range of 20% to 40%.
Great. And
that's as specific as I can get for.
Yeah. And great. Thank you so much. It looks like we're at time. But thank you so much, Dr. Alldredge, for this informative program. And thank you to all of you for participating and submitting your great questions. Please check out SHARE's upcoming educational programs and support groups, and follow us on social media. Thank you again, everyone. This concludes today's webinar. Bye.